All right. Can everybody hear? Yep, perfect. Well, welcome to the panel A of the research grant recipient talks. For those of you who weren't here yesterday, I'm Joel Watts from the University of Toronto, and my job is to moderate this panel. Well, there's nothing really much for me to do other than to introduce our fantastic speakers who have all received research grants from the CJD Foundation recently, and they're here to talk about their exciting work. We are going to have 5 presentations back to back, and so I ask that you write down any questions that you may have for the researchers, and then immediately following this we'll have a brief debrief and then a question and answer period with each of the researchers for your questions. And you can also ask questions in a more personal scenario during the moving roundtables later this afternoon. So, without further ado, it's my pleasure to introduce our first speaker, Dr. Jason Bartz from Creighton University. He's going to be talking about his work on detection of residual prions from decontaminated medical and laboratory surfaces. Jason.
Thanks, Joel. Thanks for the introduction. And thank you all for giving me this opportunity to talk about some of the work that you supported. So prions can be transmitted iatrogenically and via occupational modes of transmission. Iatrogenically, it's been shown that prions can be transmitted with various tissue or blood, also from surfaces such as surgical tools. Occupationally, and this was just talked about in the previous talk, is that there are 2 cases of occupational transmission. The best documented one was reported in the New England Journal of Medicine, where it was transmission from a stainless steel forceps via a puncture wound. And kind of based on this is that France halted prion research among safety concerns. So how does one detect prions on surfaces? So there's 2 kind of main methods for doing this. One is animal bioassay. In these studies, stainless steel wires are coated with prions, inserted into transgenic animals, expressing the transgene of kind of choice, so what you're interested in, and then the output is incubation period of disease. And this is really a powerful methodology because there's many different transgenic models that are available. But the disadvantage is that these studies take a long time and they're correspondingly expensive. More recently, the scrapie cell assay has been developed, and this is an assay that can replicate prions in cell culture, and what people have modified this assay is, again, you can take these prions that are bound to stainless steel wire, put them in the cell culture assay, and assay for prions on the surface. And the advantage of this is a relatively short duration. One of the disadvantages is that there's, at this time, kind of limited number of prion strains that this assay is compatible with. However, both of these methods, while super powerful and been really instrumental in investigating kind of ways of decontamination of surfaces, they're really not amenable to surface surveillance. And so to kind of get around this problem, we've developed a method of swabbing surfaces for detecting prions. And fortunately, I have a lot of really smart people in my group, and Qiuye is one of them. She's an environmental engineer, and she developed this technology where she could take foam swabs and swab a surface, extract the solution from the swab, concentrate it, and put this into RT-Quick, and sensitively detect prions off of surfaces. And when we initially developed this was for chronic wasting disease. Part of my group works on CWD, and we're interested in being able to detect CWD in environment. And right now, in collaboration with the University of Minnesota, we're using this technology to do passive surveillance of CWD in captive facilities in Texas right now. So for today's talk, we turned our attention to clinical and laboratory settings. And so what we did is we chose different surfaces, ones that are going to be found in the clinic or in a laboratory, so glass, stainless steel, and this can be, again, benchtops or stainless steel instruments, and then benchtop. And when I say benchtop, what I mean for the basic scientists in the group, this is the black kind of phenolic, indestructible benchtop that's in most laboratories in the world. And so what we did is we contaminated these surfaces with prions and then asked if we used various treatments, could we recover prions and detect them with RT-Quick? So in the first experiment, before I get to that, just go a little more detail on the methodology. Again, so we contaminate the surfaces, we treat the surfaces, we pre-wet these swabs, and what she found was that foam swabs are very efficient at recovering prions from surfaces. Sonication is required to disrupt the prions from the swabs, then we concentrate them and then detect them using RT-Quick. We did this with at least 3 biological and 8 technical replicates, and we were able to calculate seeding titer, which I'm not going to present today in the interest of time, but we can quantify this. So the first thing is, can we detect prions from surfaces, from glass, stainless steel, and benchtop? And what we did is did a dilution series of a known amount of prions, contaminated the surface, let the surface dry, swab the surface, and then plug this into RT-Quick. And basically what we found is that, yes, from both glass, stainless steel, and benchtop, we could very sensitively detect prions that were applied to these surfaces. So based on this, now we wanted to ask is, well, if we treat these surfaces with various treatments, what would happen? And so we used water as a negative control treatment, so this should have no effect on prion activity. We used bleach as a positive control, so there's a large amount of literature suggesting that bleach can effectively decontaminate prions. And then I'm a microbiologist, and 70% ethanol is a common disinfectant used in microbiology labs. So we wanted to ask, does 70% ethanol have an effect on prion infectivity? To do these experiments, again, we contaminated the surfaces with various dilutions of prions, either untreated or treated, extracted the swabs, performed RT-Quick, and what we found is that with water, that there was no effect, i.e. that there were still high levels of prions on the surface. With 70% ethanol, similar to water, there was no effect. Interestingly, in the prion literature, we could only find one reference to people investigating if 70% ethanol could decontaminate prions. This is from Alan Dickinson a long time ago, and as usual, he was right. And then bleach, we found a complete elimination of RT-Quick seeding activity on these surfaces. Again, consistent with the literature that bleach can inactivate prions. However, we wanted to consider the possibility, well, what if bleach is just inhibiting the RT-Quick reaction? So is this failure of detection actually from the bleach poisoning RT-Quick and not from destruction of prions? So we performed the next experiment where we contaminated the surface, we treated the surface with bleach, we swabbed that surface, and now our previous experiment that I just showed in the previous slide, there was no seeding activity. Now we took that same swab and re-swabbed with prions again. If something on that swab is inhibiting RT-Quick, it should fail to amplify. However, what we found is that they did amplify, and we had similar detection as if we didn't do this at all. So from this, we concluded that if there's any residual bleach that's coming through in this experiment, that it's not inhibiting the RT-Quick reaction, and in fact, the failure of the seeding is due to destruction of the prions and not due to inhibition of the seeding reaction. So what if the prions in the surface is not recovering? So again, we're concerned with what prions are on the surface. And so what we did is a series of experiments where we coated stainless steel wire with prions and treated the wires with either water, 7% ethanol, or bleach, and then directly put the wires into RT-Quick to ask what's remaining on the surface. In panel B, we did a series of controls just asking, can this technology work? And short answer is yes, is that our detection limit of of prions put just into the test tube was similar to the wires put into the test tube. And now in panel C, we have our negative controls, negative control wires. We failed to amplify anything, indicating that it wasn't spontaneous conversion or contamination. And then at the bottom is that when we treat these wires either with water or ethanol, we have seeding activity that's similar to the untreated wire, and then when we treat the wires with bleach, we fail to detect seeding activity. So from this, we conclude that the swabbing results are predictive of what is remaining on the surface. So next, we wanted to ask the question, is the RT-Quick seeding activity predictive of infectivity? And so to do this, we did a really straightforward experiment. Again, where we contaminated surfaces, let the prions dry on the surfaces, treated them with the various treatments, water, ethanol, or bleach, extracted the swab, and then took the extracts and split them in half. In one half, we performed RT-Quick, and in the other half, we inoculated into recipient animals to ask if there's infectivity. And so in In panel B is a series of RT quick plate controls, positive and negative controls. In panel C is the RT quick from water and ethanol, again showing robust RT quick seeding activity. Then when those corresponding samples were bioassayed in animals in panel B, these animals developed clinical signs of prion disease with incubation periods that were very similar. And then in our negative control swabs and these surfaces that were decontaminated with bleach, these failed to have RT-quick seeding activity, and we inoculated these into recipient animals. These animals remained free of clinical signs out to 300 days post-infection, and in this system, this is longer than a single infectious unit would cause disease. And then on the bottom right is a Western blot just confirming the clinical diagnosis of disease. So kind of the overall takeaway from this is that the animal bioassay and RT-quick results matched. So at least in this example is that the RT-quick values are predictive of infectivity. So lastly, we wanted to ask what effect does dust have on an RT-quick assay? And clinical and laboratory situation should be clean, but I'm a realist, and at least in the laboratory, sometimes labs are not perfectly clean. And so what we did is we did a series of experiments asking what is the effect of dust on this assay. And again, fortunately, I work with environmental engineers, so they suggested 3 different soil types that could be constituents of dust: kaolinite, montmorillonite, and hectorite. And what we did is we added increasing concentrations of these various soils to RT-Quick to find what was the inhibitory concentration of soil. So the data I'm just presenting here is from kaolinite, and in panel E, this is with 0.1 mg per mil, panel D is 1 mg per mil, panel C is 10 mg per mil, and panel B is 100 mg per mil, and at 100 mg per mil of kaolinite, the solution is cloudy, it's clearly very contaminated with dust. And at 0.1 and 1 mg per mil, we find no effect. But now as you get to 10 mg per mil, you see that that amount of dust is starting to inhibit RT-Quick, and then by 100 mg per mil, it completely kills RT-Quick. So from a practical point of view, if you're going to use this in the clinic or in the laboratory, these are some important things to know. Again, with any of these tests, it's really important to know what they can do, but also knowing the limitations of the tests, I would suggest, are just as important. So the outcomes from this work that you supported is that we can sensitively detect prions from laboratory and clinical surfaces, glass, stainless steel, and laboratory benchtop. We can activate prions on the surface, and the RT-Quick status of these surface swabs we believe is predictive of what is remaining on the surface, and it also corresponds with infectivity. And then importantly, environmental contaminants can interfere with this prion detection. So this is, again, an important consideration for those of us who are going to use this in a clinical or laboratory setting. This work and some more work I don't have time to present, this is under review right now. And while this was happening, Byron Qiuye's group published a paper in PLOS Pathogens, providing a complementary method to what I've just presented here. And in Byron's group, what they did is they took a solution and put on the surface, and then pipetted that solution off, and put that into RT-Quick, or soaked surgical tools in the solution and took that solution and put it into RT-Quick. And Byron's group, kind of being the originators of RT-Quick, they were able to do this not only with prions, but with prion-like proteins, synuclein and tau, and found very similar results to what I've presented here. So instead of kind of feeling like we got scooped on this, actually I'm really excited about this because we have 2 groups independently coming up with complementary techniques, and I think this just makes it a much more robust series of tools that we can use both in laboratory and in clinical settings. So I'm very grateful for the support of the foundation to doing the science. And as a basic scientist, I think most people assume that my most important job is to do science, but I would suggest it's one other thing, and that's training. And so from the support of this foundation, we're able to train the next generation of scientists. And Sarah Simmons, who's in the audience, did the vast majority of the work that I'm presenting here today. Sarah joined the lab as a master's student. And it became immediately clear that she was super interested in the project, and extraordinarily talented. And in the 20-plus years that I've been training graduate students, Sarah is one of the most talented students I've had the fortune to work with. And so with the support of the foundation, she's produced this work, and happily for me, she's decided to continue these studies as a PhD student, and now she's going to dive into the mechanism of how do prions bind to surfaces? How do they remain infectious on these surfaces? We really want to know at a mechanistic level what's happening. So with that, I'd just again like to thank the foundation and the families that made this work possible. I'm personally very grateful for this, not only to do this important science, but to train the next generation of scientists. This is, again, I think very, very important. In my lab, listed here are the current members of my lab. The ones in bold are the ones that contributed something to this project. So again, I'm really grateful to be surrounded by a group of really smart and hardworking people. Below are the various organizations that I work with. And again, I've been fortunate to have support from both federal and state agencies to do this work. So with that, again, thank you for your support and thank you for your attention.
Thank you very much, Jason. And just remember to write down your questions, as we're going to keep all questions to the end. Okay, let me introduce our second speaker today, Professor Parmjit from the MRC Prion unit and the University College London in England, who's going to be talking about his work on elucidating the factors required for propagating human CJD prions. Professor Jot.
Thank you, Joe. Good morning. First of all, thank you to Debbie for the invitation to come and speak. And also thank you to all of you for raising the funds to be supporting us as the researchers. But let me start even more importantly, is to thank the families in particular who actually are supporting the work that I'm going to be presenting today. So in fact, this work is sort of being done by three of us, myself, Simon, you've already heard speak a while ago, and of course, really Melissa. Wave your hand, Melissa. So, okay. So please come and talk to us. We don't know who you are, but you know who we are. So come and talk to us. We'd really like to thank you in person. Okay, with that, the research actually that I'm going to talk about, or that you've supported, has a very, very simple aim. And the simple really aim is we want to identify the factors that are required in addition to the cellular prion protein for propagating human prions. It's as simple as that. That's really all we want to do. And the approach that we're actually going to do is we want to actually look at changes at the genomic DNA. Simon told us about the genome is very important. Then he talked about RNA. We want to look at the changes at the RNA level, in fact, that take place when, in fact, cells become susceptible to variant or sporadic CJD. And ultimately, what we want to do is we want to see if there are, in fact, changes or any of these factors, are they in common between variant and sporadic? And ultimately, that all journals ask, do any of these factors have a causal role? Do they have a causative effect? Okay. Now, before I do that, I need to give you a little bit of background. And the background is, in fact, when I joined the unit about 20 years ago, many moons ago, I actually inherited at the time, was a very simple project. And in fact, it was so simple that as someone who actually didn't know anything about prions or didn't know anything about neuroscience, Professor Collins said, well, why don't you try and make us some cells that can propagate human prions in a dish? A very simple project. However, the catch was that we actually don't know what is required for propagating prions. And that was the catch. But he thought, well, I've spent my life doing cell engineering. My background is cell molecular biology, and I make cell models to study problems, and I'd been studying them in the context of cancer and aging. Well, you can do this. This is only prions. So essentially, what I decided was that, I don't know whether you can see the blue, I'm afraid. So what I decided was a very simple approach. So in the prion field, when people wanted to make, or even our unit and others in the field wanted to make mouse models of human disease, what essentially was done was to take mice in which, in fact, the endogenous mouse prion protein had, in fact, been deleted. And these mice were then essentially reconstituted back with the human protein. And in fact, this is how these human models were developed. So I thought, well, why don't we do that? And essentially, it's taken us 20 years, in fact, to do that. And what I like to tell you is what's been successful. And the approach that we've used, in fact, Joel has, in fact, also used in his lab to develop models for propagating hamster prions and also prions from bank voles. And Schatzel's group, in fact, in Canada, actually have done that for CWD. And unfortunately, you can't see everything, but so the approach we've done is we've taken cells. For example, in this slide here, as we're using CAD5 cells, it's actually a mouse brain cancer cell line, in fact, which propagates mouse prions. And essentially what we've done is we've silenced the endogenous mouse prion protein, and we've done that in a number of ways. So originally we started doing it by RNA silencing because, in fact, in 2005, CRISPR-Cas had not yet been discovered, so what we had was silencing by RNA interference, and we also subsequently have done it with CRISPR-Cas. And what we then do, in fact, is we reconstitute these cells. So before we do that, one of the things we need to do is we need to make sure that all of the endogenous protein, in fact, has been deleted, and then we reconstitute these cells, in fact, with the human prion protein. And what we've, in fact, done is we've used either the methionine variant at amino acid 129, or we've used the valine variant also at 129. We've done a couple of other tricks, which is, in fact, to replace the signal peptide from human to mouse, so, in fact, they express very well on the cell surface. And so, essentially, we've developed now cells which are, in fact, essentially recapitulating what was done to make human models, so mouse models of human disease. Then the next thing, in fact, we've done, since our previous speaker mentioned to you a scrapie cell assay, we've actually developed a human counterpart of that assay where we take, in fact, sorry, didn't mean to do that. If I can do the pointer, we start with brain tissue, we homogenize it, and then, in fact, what we do is we take this tissue and we challenge susceptible cells with that tissue. And essentially what you then have to do is, because prions are very, very stable, they don't turn over, what you then have to do is you have to passage these cells a number of times to dilute out the inoculum. And then what you can do is you can take these cells and then you can actually look to see whether, in fact, you're now making de novo prions. So essentially, this is the human prion assay. If anyone would like more details, I'd be happy to talk about it afterwards. So what we've done, or what actually Melissa has done, this is a bit of a busy slide, we've actually taken this approach and we've actually challenged, so what I'm going to focus on today is variant CJD. We also have done this for sporadic, and essentially what Melissa, sorry, gone too far. Okay. So what we've simply done is we've taken these cells, we've reconstituted them, we've actually challenged them with variant CJD inocula. In fact, these inocula come from the patient, from post-mortem brains. And so we've actually have used a number of patient brains because we didn't want to isolate cells which would be specific for one brain type because we don't know how many strains there are. So we basically have used multiple brains. And what we were able to do is actually identify a starting population which actually were susceptible to around 10,000-fold dilutions of human brain. Now, it has to be this level because if you use any more concentrated, there's so much debris, in fact, it actually kills the cells. So this is, in fact, the highest concentration. And in fact, at this point, Melissa took over. And what she was able to show, in fact, that these cells actually will respond to a number of human variant CJD brain homogenates. And then what she was able to do was, this actually took about 4 years, was able to actually single cell clone and then in fact show that these cells in fact, so let me back up a little bit more. The way, this is a mouse cancer cell line, and what actually mouse cancer cells have, or all cancer cells have a property, is that their genome is unstable, which is to say that in fact that at every generation they can throw off variants. So essentially what Melissa did was take this first round of cells, which we knew were susceptible, dilute them out, look at 500 different clones, and look for a clone, in fact, which was still susceptible to those variant inocula, and was looking for to see if they were becoming more and more sensitive. And what I want you to note is that at the end, that these cells, in fact, are now susceptible to 10 million-fold dilutions, whereas we started off only at 10,000-fold. So essentially, by going through the serial single-cell cloning, we were able to identify very, very susceptible clones. So while we were doing this, one of the first things, in fact, the director asked us to do was, well, show at a very early stage, are you actually propagating variant CJD? And to do that, our previous speaker talked about mouse bioassays. We've set up two sorts of mouse bioassays, either in wild-type mice, or I'm going to show you also in humanized mice. What I want you to note is that, in fact, on the left panel here, this is end-stage mice, wild-type, with the original inoculum, and what it's actually showing is histopathology, and in fact, what I want you to look for is this coloration of brown, or this color here. This is the original brain inoculum, and this is, in fact, this cell passage inoculum. What I hope you'll now see, in fact, is that the pattern of expression is exactly the same. And this is the control on the right to show, in fact, it's not just a residual inoculum. So this is, in fact, one brain sample, and we've done it with a second brain sample. So in here, in fact, once again, so this is demonstrating that actually we're propagating bona fide variant prions. Of course, this is only wild-type mice. You might say, well, what happens, in fact, in humanized mice? We've also, I don't want to do this. So in fact, we've also done that in humanized mice, same approach. On the left panel, the original inoculum. On the right is cell passage material. On the right is controls. What I hope you'll see here, in fact, this is a schematic. This is the expression. This is, in fact, actually the same as before, cortex, hippocampus, midbrain. And so what I hope you'll see here is the pattern of expression is once again exactly the same. In addition, what I hope you might see here, in fact, is that we now get florid plaques because these are humanized mice. Okay? And in fact, we've done that we get the same result. Sorry, we jumped too far. With multiple inocula. Unfortunately, I don't think this is going to be visible. So what I now want to show you in just a couple of more slides is that in addition to demonstrating by mouse bioassay, we can show, in fact, that these later stage cells, in fact, are susceptible to increasing dilutions. I don't know whether you can actually see single spots here, but as the cells become diluted, the spot number goes down. And this is basically to demonstrate you can do a dose dilution. And this is, in fact, to demonstrate that the cells we've developed with the variant approach, in fact, they're also susceptible to homogeneous made from heterozygous CJD patients. And in fact, what they don't do is they don't actually respond to sporadic disease. So the cells actually we've made, they're propagating bona fide variant CJD, but they will not propagate sporadic. Okay, so, and what I haven't shown you today is that we've also made cells that propagate sporadic. We've shown by mouse bioassays that they're propagating, in fact, bona fide strains, but those cells, in fact, do not propagate variant. And this is, in fact, the type 3 MVVV strains that we're doing, okay? In addition, I'd better not show this. What we've also done is, in fact, we've used the human inocula to, in fact, develop chronically infected cells. So we can do this now. We've done this with multiple homogeneous from multiple patients. And these chronically infected cells, in fact, can be frozen, can be rethawed, and they actually maintain they maintain infectivity. Furthermore, in fact, these cells, in fact, can be cured. We can cure them with ISM-18 antibody, which is an antibody against the anti-PrPSc. We've actually tried curing them with some of the small molecules that people have talked about. They do not cure these cells. So clearly, one possibility is we should go forward and we can do small molecule screens to see if we can identify small molecules that can specifically cure these human strains. And we've done the same for sporadic. Now, the project that you really funded me to do was to ask, in fact, can we identify, what I'm now telling you is that we've actually developed cell models for variant as they become more and more susceptible, same for sporadic. What we really want to now ask is, are there any changes at the DNA level that take place that correlate with, in fact, the cells becoming more susceptible, and also the RNA level, both for variant and sporadic. So what, in fact, we've done is that we've actually extracted genomic DNA from each and every one of these cell lines, and also we've actually extracted RNA from each of these cell lines in quadruplicate. And all of these, in fact, have been sequenced at a very high level of sensitivity, and in fact, analysis of this data is now underway. And I hope, Debbie, if you'll invite me back in a year, I'll come back and tell you what are the specific changes, in fact, that take place. But in the last couple of minutes, I'd like to share with you a little bit of data from the RNA-seq experiments. What I want you to hear, and I think the reason why I show this, it looks very interesting. So this is a machine learning, principal component analysis is a way of looking at the RNA-seq data to see how similar is the data from different cell lines. And the reason why I'm showing this is the following. If you look on top here, this is the parental CAD5 cells that we started with that actually are propagating mouse prions. This grouping here, in fact, is the same cells when we knock out the mouse prion protein. I want you to note to see how far the cells change in terms of RNA expression. And what we've then done is these three groups here, in fact, is where we've now put back the human prion protein. There's one grouping here, and one here. The only difference between these three groups is that this one is susceptible to variant, and these two are not susceptible. This, in fact, corresponds to the next generation susceptible. These two groupings here correspond to the next generation, so on and so on and so on. What I want you to take away the message is that as the cells are becoming more and more susceptible, there is a gradual increase and they move in a very sort of systematic sort of way. So this was with variant, and we can do the same with sporadic. This is the original cells. This is the cells when they've been silenced. And this is, in fact, as the cells are becoming more and more sensitive. So essentially what we're now trying to do, in fact, is which is Simon and Holger who works with him, is using bioinformatics and machine learning to try and identify the genes that are responsible for these changes. And furthermore, we'd like to identify the pathways, in fact, that are changing, and ultimately can we modulate them and ask if they have a causal role. And then we can cross-compare between sporadic and variant and to see, in fact, whether there are things that are in common or they're indifferent. So I'm going to jump the last slide, which was dendrograms. But to go this, in fact, this is a summary of what I've actually tried to tell you today, which is that, in fact, we've developed dividing cells that are susceptible to both variant and sporadic. We developed a human prion assay for infectious vCJD and sporadic CJD. And this is, in fact, the type 3 MM or VV. These cells, in fact, propagate bona fide prions. There's no switching of strains. Basically, they're propagating the same strain that we started with. This was very important to us. We've also developed chronically infected cells, in fact, that remain in infectivity upon freeze-thaw. So, in fact, we can take an inoculum for any patient, variant patient, we can actually develop chronically infected cells within perhaps no more than 4 to 6 weeks. So these cells, in fact, can then be used. They can be cured. And, in fact, we're actually doing RNA-seq and whole genome sequencing to try and identify if there are any changes that specifically correlate with the increases in susceptibility. I'm afraid it's only taken me 20 years to get this far, but it was a very simple problem that I was given. But last but not least, in fact, is in addition to Melissa, who's really done most of the work, there are a number of other people, in fact, who've contributed. But thank you for listening, and I'll try and address any questions during the roundtables. Thank you.
All right, thank you very much, Dr. Jat. The third speaker is Dr. Qiuye Li from Case Western Reserve University in Cleveland. He is the first recipient of the Postdoctoral Research Award offered by the CJD Foundation, and today he is going to be speaking about his work on high-resolution structure determination of MM1 and MM2 sporadic CJD prions. Dr. Li.
Thank you for the nice introduction, and thank you for inviting me here. My name is Qiuye Li. I'm a postdoctoral fellow in the lab of Doctor Service at Case Western Reserve University, which means I don't run the lab myself. So I'm especially grateful for the opportunity to have this funding so I can conduct some very exciting exploratory work during this 1 year duration. So I would say, as a very simple-minded chemist by training, my aim of this research project is even simpler than the previous speaker, I believe, which is guided by the great physicist Richard Feynman that you simply look at the thing. In this case, we want to look at prions. And if you remember the presentation yesterday, that prions are pretty much protein filaments who behave like transformers, which means they have two roles. One is making damage to our brain, which means neurotoxicity. The other is structure replication, which you can think about the transformer is trying to convert other Chevrolet Corvettes or every single car into this transformer form, which is a lot of damage. In real world, the prions, they are typically show as rods or filaments or amyloid, which means they have this linear shape, but there are also other variants people have been studied think they are also infectious or pathogenic. But in this work, we will mainly focus on this filament-shaped prions, which I will quickly go through this process, how they form filaments. And we all know the protein usually, they are happily behave as what they are, as the normal folding, but in very, very rare event, they will become this misfolded form. And then this misfolded form is usually unstable. It often requires multiple copies of misfolded form to bind together to stabilize this wrong folding of this protein, which takes a lot of time. And this long nucleation phase correlates very well with the late onset of many neurodegenerative diseases, including prion diseases. But once such a nucleus forms, we'll quickly convert other good Chevrolet Corvettes into transformers, unfortunately, and this will happen very fast. Eventually, these mature fibers will occupy all of your brain. So to look at those mature fibers, we need very powerful tools, nothing like magnifying glasses, but more like a microscopy. So here is a very conventional method by staining prions with specific antibodies and look them under microscopy. And you can see those very dense things within the neurons of your brain. And then we can also go into higher magnification using transmission electron microscopy or atomic force microscopy, which gives you more details of those filaments. And sometimes you can get some 3D information by very precise mechanical tapping. And last but not least, in recent years, when you collect several thousands of very noisy images like this, and then you overlapping them, reconstruct them, you get a very sharp image. This is possible by recent development of cryo-electron microscopy. And not only this very sharp image you can also get the shape of this cross-section. You know how protein misfold and become this filament. So why this is important? Because prion disease is a very specific case of neurodegeneration because it's solely caused by a misfolded form of protein or amyloid. Unlike Alzheimer's disease, Huntington's disease, or Parkinson's disease, that either the prions or the amyloid is not directly connected to disease, we can account the neurodegeneration simply to the existence of prions in most of prion diseases. So once we know the structure, all the pathogenic features are actually encoded in the structure of a protein. So this is kind of a perfect example for structural biology studies. For example, we originally proposed to compare the structure of MM1 and MM2 prions, which we can understand the difference between those two subtypes. And once we know the structure, we can possibly design more precisely targeted diagnosis and therapeutics as the basis of modern pharmaceutical science. So actually, this is not just a proposal, but it's happening for some studies, such as tau filaments in tauopathies. The tau filaments is very well known to exist in neurodegeneration like Alzheimer's disease, and the recent progress of cryo-electron microscopy actually determined the high-resolution structure of tau filaments. Here I'm only showing the cross-section of those filaments. And then some groups have started to design new drugs and diagnoses using these structures. And that effort actually includes one of mine that we found a case of rapidly progressive AD, which was mistakenly identified as some form of prion disease because the patients only survived less than 1 year after diagnosis. But we do find that these are actually a specific form of Alzheimer's disease, and the tau filaments extracted from the brain of those patients have some extra decorations, if you can see, pointing by those red arrows. Whether those extra decorations are related to the rapid progress of this type of AD or any type of rapidly progressive neurodegeneration, we need more evidence. But obviously, this is a great start to understand the disease in a molecular level. But for prions, even though the connection of the misfolded protein and the disease is much more clear, we do not have much information regarding the high-resolution structure. Here are the example of several rodent prions which are passed from scrapie. And it's probably not so surprising that they differ very little from each other, considering they all have the same original source. So we're trying to dive hard into the real details of actual human prions using this method of cryo-EM, which started in 60s last century, but really got a huge boost recently and won the Nobel Prize in Chemistry in 2017. The basic idea is to get a highly purified sample of prion and deposit as a very thin layer on the metal mesh, and then you quickly freeze it and the image using this roughly 10 feet tall microscopy, getting 2D images, and then use some computational method from the 2D image, you can get a 3D structure of this protein. Obviously, this is a globular protein, but you can also do it with filaments. So this usually takes months of, if not years of work. Most notably, you need to spend a significant amount of time to purify this highly purified protein. As well, you have to do some computational work. Most often, you are using something called GPU, which is the same thing you use to raster a 3D image when you're playing video games. But here, we are trying to get a 3D image of a prion filaments. So here is our effort in purifying prion filaments from 6 cases, 3 from MM1 and 3 from MM2. This is a very busy gel, but it just confirms we do have those 2 subtypes in those patients. And then we adapted and modified an existing protocol, which was originally designed for purifying prion from rodents. Actually, we realized that the tissue of lab animals and human differs a lot, so we have to modify significantly to get this purification. And then after purifying the human prions, we will deposit on a very smooth surface. And then we touch them using this very tiny probe. And then by amplifying using this laser beam, we will be able to get this surface feature, which will tell you what what our purification looks like. And here are some images from 2 of the MM1 cases. Unfortunately, you won't see much, but you probably can find some fibular structures, very faint in the background, which means you have a lot of impurities that are much larger than prions, and then you barely see the good MM1 prions. And then we move to MM2, which in my eyes, they are significantly better. But I can find this audience may find it difficult to see, so I will give a more enlarged image. So you will see those fibular structures accompanied with those large sphericals. And those fibular structures are actually what we are really interested in being human MM2 prions. So to summarize this effort, we realized that tissues from actual human patients are very different from lab animals, and current purification methods are heavily designed for lab animals, not necessarily because we are interested in mouse prion diseases, but most because we have a lot of chance to modify the method because we have almost unlimited supply of mouse brains. So it's obviously very important to get highly purified prion filaments to carry on this work. So within this short period of time of 1 year, we turned to a different type of prion, which is the CJD. Actually, I noticed that this audience has a surprising high interest on this prion disease. So I'm glad I bring it up here that these are generated by our collaborators from deers, but transmitted to deer-raised mice, which means when we do the purification, they will come from those lab animals, which makes it easier for us to do the purification. So here are a good CJD prion images. What we are trying to get for human prions is just highly purified filament structure like these. And then if we got such an image on AFM, we should be able to get a good cryo-EM result. And here shows how a cryo-EM image looks like. They are not as sharp and nice as those AFM images, but they do preserve a lot of high-resolution image after some image processing. You can see we do overlay those images together. We have a much sharper and more detailed image, somehow resemble those blurred image on the top. And then if you look into more details, I will enlarge part of this filament. You can see those lines perpendicular to the axis of the fiber, which are actually layers of beta sheets. And then what we are trying to do in 3D reconstruction is to understand what the cross-section looks like for each layer, although they are all identical. And here is a small image showing you what this cross-section looks like. I didn't put it at the corner on purpose, but only because these densities are actually off-center. You can see from this very wavy shape of this CJD prions. And you can also see that the so-called twist is relatively slow comparing to many other amyloid, which give us a hard time to reconstructing the structure. But if we do a little bit of a comparison, I hope this video will play. Okay, good, they do spin. So here are 3 cases of prion from different sources, GSS prion, RML prion, and as well as those CJD deer prion. And you will find that they have different shape and different cross-section appearance. Especially this CJD prion is quite unique with this off-centered position as well as this curvy shape. Whether these are related to different pathogenic features or different infectivity, whether that can be passed to human beings, probably one of the things we care about most, will require a more detailed inspection, especially we have to boost the resolution a little bit more from this reconstruction. So just to summarize, within this short 15 minutes, I actually talked about 2 stories. About human prions, we realized how difficult it is to purify them, especially for this MM1 and MM2 cases. But we do find that MM2 prions have a little bit more fibular content than MM1 prions from this very short group. And then we did a little bit more study regarding those CJD prions, which are quite different than those well-studied mouse prions. And we hope this kind of work can lead to some future designs of therapeutics as well as diagnosis. Last, usually I will thank the funding agency at the very beginning, but I think for this kind of work, especially after I realized how difficult to purify human prions, I would like to thank the patients' families first. Without this donation, it's not possible for us to conduct those trial and errors. Thank you. And then I would like to thank my lab mates, my advisor, as well as our collaborators to make this project possible. I would like to thank the National Prion Disease Pathology Surveillance Center for providing us those human samples, as well as the foundation to sponsor this work. I guess I will not able to answer questions as of now, but please do make notes of your questions. Thank you.
Well, thank you very much, Dr. Lee. And our next presenter is a fellow Canadian, Dr. Robert Mercer, currently at the Boston University School of Medicine. And he's going to be talking about his grant that was entitled The Identification of PrPSc Interaction Partners and the Characterization of Their Role in Prion Infection and/or Propagation. Dr. Mercer.
Thank you, Joel. And thank all of you for coming to this talk, and thank you for inviting me to speak. I've been looking forward to coming to this meeting since I heard I was coming. I'd like to start with my acknowledgments, because I'm always fearful of running out of time. This work that you funded isn't funded by anything else currently, and so your contributions through your hard work, through the Strides for CJD and through the foundation grant, and contributions in the memory of your loved ones, it really makes all of this possible. So what is it we're trying to do? Like everybody else, I say it's simple, but I guess it ends up being much more complicated. But what we really want to do is apply a new technology called micromapping to discover proteins that are interacting with PrPSc or with prions. This is highly complementary to some of the other work you've heard where people are looking at genetic changes that contribute to susceptibility to prions. But here, we want to look specifically at a protein-protein interaction. And then once we identify these things that we call hits, we want to interrogate them using methods like CRISPR to remove their expression, or other systems where we overexpress the protein to see how the loss or gain of more of that protein impacts the susceptibility to disease. So, of course, the number one interaction of PrPSc is absolutely required for disease is PrPSc. And without PrPSc, there is no prion disease. And we've known this for more than 30 years after the publication of the first prion knockout mouse. So this animal is completely resistant to disease. And since then, cows have been developed without prion protein, and recently goats were discovered that are naturally devoid of the protein, and they too are resistant to diseases like CWD. But it's more complicated when you look at a system that also, or rather when you look at a system that's expressing PrPSc. So what we say is PrPSc is required for disease, but it's not sufficient. And what I mean by that, I think I'm hoping I'll be able to get across to you in this slide. So here in the top, in A, we were just simply looking at the expression level of PrPSc in a number of different cells. And in B, we've infected these cells, and then we're looking at prion levels. So we're ignoring PrPSc and just looking at prions. And what you can see here in the blank sections, these are cells that cannot be infected with one of these given strains, despite the fact that they're expressing PrPSc. This also extends in vivo in the brains of some animals. So in this particular study, you can see the expression of PrP in this kind of purple-blue color, and using the same three strains, we see that the prions deposit with different intensities in different regions of the brain. In particular, I'll point out here the cerebellum in the very back. Only one strain deposits there, despite the fact that the prion protein is expressed in that region, even when challenged with these strains. So what's going on? Like something you can take this to tell you that something else is controlling the susceptibility of these regions. It's not simply the expression of PrPSc. And some work has been done in the past to identify regulatory networks that are associated with susceptibility or resistance to infection. And so we wanted to come at this question anew using interactomics. And so interactomics is really just looking at the entire map of interactions of a single protein. So when we say omics, we really just mean all. So all interactions. And we've done, or I haven't, but work has been done looking at the interactome of PrPSc. And we've learned quite a lot from that. And so how these are done, they're done with intact cells. And then here you can see the prion protein in a rainbow color, and these are just sort of cartoon proteins here that aren't any particular protein. And in these sorts of experiments, we cross-link the proteins using formaldehyde, so now they're permanently stuck together. And then we isolate the prion protein using antibodies, and anything that's stuck to the prion protein is going to be isolated with it. Then they're analyzed by mass spectrometry, and these instruments can give us sort of like a protein fingerprint. So any protein can be identified using a mass spectrometer. And then we can build protein interaction networks. So we have our protein of interest in the middle, and you can identify all the proteins that were pulled down with it. And this has been highly successful, and there have been a number of papers published looking at the interactome of PrPSc. And so the old adage that, to understand a person, look at the company they keep, is true also for proteins. And so the knowledge of these interaction partners of PrPSc has led to biological insights into the function of PrPSc. And over the years, there's been many papers published looking at the function of PrPSc. And this was a particularly high-profile paper that showed that the PrPSc was interacting with the A-beta peptides in Alzheimer's disease and mediating some of their toxic effects. And so why can't we just do what we've done with PrPSc with PrPSc? And the problem, and the reason we can't do that, is because while PrPSc and PrPSc are very, very different structurally and biochemically, they're the same protein. The chain of amino acids from beginning to end is identical. And this makes the traditional techniques that we would use for analysis of such an interactome unsuitable for PrPSc. And this is where micromapping comes in. It's a novel proximity labeling technique that was published in April 2020, right at the outset of the pandemic. And perhaps then it wasn't a surprise that it was very quickly used to look at the interactions of SARS-CoV-2 with cells. And there's been a couple of papers looking at those sorts of interactions using this method, and even interactions between one cell and another. So how does it work? We'll go back to this cartoon of the cell surface, and here's the prion protein again in rainbow. And the first part is going to be somewhat familiar, but then the novel part comes in. So we target the prion protein with an antibody, and then that antibody is targeted with this catalyst labeled antibody, and this catalyst is really the star of the show. So when you then put a non-reactive probe into the system, this probe is benign. It doesn't do anything until it gets hit with energy that is released by this catalyst when blue light is shown onto the cells. And what's interesting about this energy release is that it's only effective within a very small defined radius from the catalyst. And when these probes interact with this energy, they become reactive, and they permanently bind to any protein that's within this sort of dotted circular area. And that allows, and then we can isolate the proteins based on them having this probe attached to them and identify them by mass spectrometry. And so as a preliminary experiment for looking at PrPSc, we went ahead and did this for PrPSc. And we were very happy to find, so this is the list of the top interactors that we identified this way. And we were very happy to find here in bold a number of proteins that have already been identified as PrPSc interactors using other methods. So this tells us that micromapping is going to be useful in our cellular systems. And so we want to turn now and do this with PrPSc, but we have to modify the method a little bit. So instead of using antibodies to detect the PrPSc, we are labeling the prions themselves directly. And by doing it this way, we would then ignore any contribution to PrPSc in downstream analysis, and we'd only be looking at things that were in proximity to the prions themselves. But there's a number of things we had to do before we could do these experiments. PrPSc is notorious for losing infectivity if it's treated a little too harshly. And so first we wanted to know and make sure that these catalyst-labeled prions were still infectious. And so we did a direct comparison of purified prions that are not labeled with prions that are labeled. And in the middle here, we just have a mock infection, which is essentially uninfected cells. And when we analyze these by Western blot, again, looking only at the prions and not the PrPSc through digestion with this enzyme PK, we see that the catalyst-labeled prions, we get the same sort of signal intensity. And so the answer is yes, they're still infectious, and we can move forward with our experiments. And so the next thing we wanted to do was compare the labeling efficiencies between the antibody-based method that I showed you first and this prion-based method. And this is important because when you're doing these sorts of experiments, you're always worried about your signal-to-noise ratio. And you know, if this is a new concept, you may be familiar with it if you're ever driving down the highway and your music's too loud and you can't hear the police siren behind you. It's because you have a bad signal-to-noise ratio. And so when we compare both methods directly, here's a Western blot, and this time it's not for prions, but it's for this actual probe that we've used. It's called biotin. And you can see this streak of dark signal, and that just represents all the different proteins that have now been bound by this probe. And so they're generally equivalent. I've coded them here. The antibody is this lane, the prions are this lane. And so this tells us that the efficiencies are roughly equivalent, and we can proceed with our experimentation, which is exactly what we've done. And so currently, we're doing micromapping using immortalized cells that are susceptible to prions, N2A and CAD5. And we're using multiple prion strains, because if you recall, in an earlier slide, I showed you that using 3 different strains, that different cells responded to them differently. And we hope that by looking at the interactome of each of these strains individually, we can sort of piece together a puzzle of how each one may be interacting with the cell. The lab has a lot of experience using cultured neurons, so we would move from immortalized cells into these systems, and then eventually even using brain slices directly. And as far as interrogation downstream, this is, it's not important what these are, but this is just to show you. You can see on this Western blot here, there's signal, and then the signal goes away. We've got a really great CRISPR knockout pipeline, and so we can interrogate any hits we get up here through methods like that. And hopefully we'll identify some novel drug targets this way and learn a lot about prion biology as we do it. So in summary, identifying the interaction partners of a protein is absolutely critical for understanding its function. Absolutely critical. And we've spent a lot of time adapting this micromapping method for use with prions, with PrPSc. And this is really all of it in a nutshell. We're going to use this technology to investigate the interactome of multiple prion strains and multiple cell types to identify other proteins, of course, besides PrPSc, or even other cell components that are really critical for prion propagation or infection. And then I'll go back to my original acknowledgement slides and just acknowledge my collaborators at Boston University, David Harris, LaDonna Min, and John Gatula. And at the University of Toronto, we're working with Gerold Schmidt-Ulms, who is a brilliant mass spectrometrist with a long-standing interest in PrPSc interactomics. And thank you for your kind attention.
Thank you very much, Dr. Mercer, and I'm pleased to announce that we are right on time. And so last but not least, it's my pleasure to introduce Dr. Valerie Sim, my Canadian colleague from the Centre for Prions and Protein Folding Diseases at the University of Alberta in Edmonton. And she will be talking about her work on the contribution of oligomeric prion populations to phenotypic heterogeneity in VPSPR versus silent prions. Dr. Sim.
Sorry for the title. I have to first decide whether to wear glasses or not. I'm approaching that age. Anyway, thank you. It is a pleasure to be here. I've been to these conferences in the past, and as others, my colleagues have echoed, it's a privilege, and I'm always humbled by your support, interest, and devotion to our research. So, without further ado, see if I guess the right button. This is a better title, perhaps, for the talk today. So, the origin of prions. Sure, that's got to be easy, right? As the theme suggests. So, come with me on a little bit of an exploration of some ideas of where on earth prions may come from. And yes, come on in. The remnants from the last group. So, why? This is a question I get often from my patients. So I'm a clinician scientist, and when patients are first diagnosed with this disease, this is a common question. Why? Why did I get this? Why did my loved one get this? And I don't have a lot of good answers. I can reassure them somehow. Well, it's spontaneous. You didn't catch it anywhere. It's 1 in a million. You're just unlucky. But those are really unsatisfying answers. Where does this disease really come from? Why does it start? So I think you've been introduced over the past couple of days to what I call the prion life cycle. So I'll just, again, go through relatively quickly. This starts with the normal prion protein, which lives on the cell, it's folded, and hopefully is behaving itself. But then something happens, a conversion event where the shape changes. And we don't know what that shape is at the start. But whatever happens, it propagates, it templates itself onto its neighboring protein, causing it to convert. And then it builds up in the cells, it spreads through the brain, and then causes holes to form in the brain. Spongiform change, because it looks like a sponge, causes neurons to die, brain cells die. Astrocytosis, so one of the cells in the brain that starts to get reactive, and you build up misfolded prion protein. So everything in brown that you see on this slide, that's the misfolded prion protein, sometimes called PrPres because it's resistant to enzymes, or PrP-scrapy because it causes disease. And a takeaway point here too is each of the patterns you see is different. The brown staining, the brown pattern's different because there are different strains, different subtypes of disease. And as we've been hearing this morning too, shape matters. And we have different shapes of misfolds that cause different types of disease. And here's just an assortment of over the past few years. It's been very exciting to see the progress in solving these structures. You know, from AFM to cryo-EM and these attempts to really understand what is it that's building up in the brain. So the top three here are some rodent-adapted strains. The bottom two are human strains, the GSS. And as we've heard, work is underway to try to solve more structures of the human strains themselves. However, while these are very exciting, these are capturing the end product, the large fibrils, because that's what you can isolate from the brain. That's what you have. So that's, like you say, who was quoting Feynman? You know, you look at the thing. Well, that's the thing we can find. That's the thing we can see. So that's what we're looking at. But is that all that's there? How does it start? We go back to the beginning. So what does this first misfolding event look like? I think it's important to recognize that the research models that we use are not spontaneous. You know, we could take 1 million mice and wait a bunch of years and hope one of them just happens to develop the disease. Well, first, no one's going to fund that, and it's just not a doable thing. So we infect our animals. We use diseases that are already ongoing to try to understand this. But it doesn't get at the question, what triggers it at the beginning? If you don't inject the brain with prions, why? How does that start? And when something misfolds, does that necessarily lead to prion disease? So we're making proteins all the time. We're made of proteins. And you know what? We don't always get it right. We're often misfolding our proteins. Fortunately, there's evolved a lot of systems to get rid of the misfolded proteins when that happens, so we don't get a variety of diseases. So that raises the question, does every misfolded form of the prion protein lead to disease? Well, we know that's not true because even in the lab, when we take the prion protein in a tube, you can shake it up and you can make it turn into all kinds of beautiful fibrils. But they don't cause disease if you inject them into an animal. So we know there are shapes that don't cause disease. So misfolding may happen, but what is it about the shape that causes disease? So how do we address this question? So we thought, well, do we know anything about prions that are the wrong shape, that exist, that don't cause disease? And can we compare these to shapes of prions that do cause disease? So I want to introduce this idea of these silent prions. So it turns out that in normal brain, you actually can find trace amounts of misfolded prion protein. And this has been dubbed silent prions. Now these are at very low levels, and they look a little bit different than the typical disease prions, and importantly, they don't cause disease. So the question is, what's different about this misfolded prion compared to the infectious ones? And is this the starting point for prions that become infectious? So what we did is we tried to look at prions that shared similarities with silent prions, and to compare the size, the shape, their stability with the silent prions and see why one lies dormant while the others cause devastating disease. So to do this, I want to introduce one of the techniques that we're doing, which I'll call fragment analysis. So if you think about it, when a protein misfolds, parts of them are tucked away so they can't be cleaved by enzymes. So imagine I take a rope and I tie a knot in the rope, and then I'm going to take scissors. All right, so I cut the rope in these two places, but I can't cut the knot. Now I unfold everything that's left, and I get a couple small fragments and a big long fragment. That tells me something about what the knot was, where it was, how big the knot was. So now imagine I have, a lot of knotty ropes, all right, different knots. And when I try to cut these with scissors, I get a bunch of different fragment sizes. All right, so now I've chewed everything up, chopped it up with scissors, and I've got a bunch of different fragment sizes. Well, now I can separate these fragments by size and try to see, okay, how long are the different fragments? What does that tell me about where the knot was, or how tough the knot was? Well, it turns out, if I show you, you've seen a few of these now, Western blots. You can't go to a prion conference without seeing our favorite blobs on Westerns. Well, that's what we're looking at. We're looking at proteins that are different sizes on a blot. Now, this is the most common appearance. You'll see lots of these 3-band patterns if you go to a prion talk. Well, it turns out the silent prions look like this. So this is normal brain homogenate, and with the arrows, you see these multiple fragments. So at least 4 to 5 different fragments are here. And you might say, well, that's totally different looking than what you just showed me. True. However, here are some disease-causing prions, rare subtypes of prion disease, and look at their fragment profile. All right. In fact, in a couple of cases, they exactly overlap with the silent prions. So let me introduce you to a few of these rarer of the rare prion diseases that overlap silent prions. So the first is this variably protease-sensitive prionopathy that some of you may have heard of. It's actually was discovered and named by Gambetti's lab. And this is a spontaneous form of disease. It's very rare, 1 per 100 million. It affects older patients, slower disease course, but it has the same fragment overlap as these silent prions. So does something happen to these silent prions that then accelerates them or allows this really rare syndrome to evolve? Don't know. Then there's a genetic form, V180I, same fragment overlap. However, it's a genetic disease, but it's not 100%. It only confers about a 1% risk. But it too, older onset, slower disease course, and it's been proposed that maybe it's actually the same structure as VPSPR. One caused by a mutation, the other spontaneous. Could both of them be influenced by the buildup of silent prions? And then as another case to compare, we wanted something that had a similar fragment profile, but a little bit different presentation. A117V, all patients develop the disease, 100%, but it affects much younger patients, and again, it's a slower disease course. So these were the ones we chose to look at. So quickly, I'm going to show you some of the data. The first thing we did is say, what are the sizes of these particles? They build up in different aggregate sizes in the brain. So we have a technique in my lab that's called asymmetric field flow fractionation, because we're all about long words, or AF4, we'll say. So we separate the particles by size. When we compared the size distributions of prion particles in these 3 disease-causing cases, the striking thing here was this A117V was different. You might think, okay, well, it had different fragment sizes. It affects younger patients, so yeah, it makes sense it would be a bit different. It had more small particles, whereas the other one, V180I, or VPSPR, had larger particles and looked very similar. Maybe they are the same disease. Then we wanted to say, well, how stable or how resistant are these fragments if we try to digest them? So imagine I'm back at the rope with a knot, and I'm taking scissors, but I start to get more and more and more scissors, and I just go at the knot. Eventually, I will get through the knot. So this tells us how knotty the knot is. I should have named it Naughty Knots. That would have been a way better title. Oh well, next time. So what I'm showing you here, as we increase the concentration of enzyme, you see the signal goes away. And this tells us how resistant that knot is. And again, there are some differences appearing. A117V is the least stable, and V180I is the most stable. With VPSPR is actually kind of in between. And in fact, when we look with different types of antibodies to probe these fragments, we find that indeed, these two appear to be different after all, not necessarily the same type of disease. And we've gone further to actually say, if we look at the specific fragments themselves, are they varying in how resistant they are? And the answer, in a nutshell, is yes. So the graphs on the bottom, basically, if you compare orange to green, blue to red, or what is that, purple to clear, between V180I and VPSPR, the fragments themselves show different stabilities, suggesting that these two are not the same disease. They have the same fragment pattern, but they don't have the same properties. So that's helpful. So how does that tell us anything about the origin of prions? Well, imagine we have the scenario. We have normal prion protein. We have an unstable form, one that's sensitive to protease, we'll say, to the scissors. And we have an unstable form that is causing disease, and then we have a stable form that's causing disease. So at some point, we have this conversion to an unstable form and this buildup of silent prions. And so far, when we've looked at our silent prions, they're in very low amounts, but they appear to be very small in size and sensitive to protease, relatively unstable. And we thought, well, do these therefore share any resemblance to VPSPR? Well, it turns out they don't really. The same fragment sizes are there, but these ones have a much larger particle size, and the stability is they're stable, more stable than the silent prions. And the V180I mutations are also different from VPSPR. They have the same fragment pattern, but these ones are actually a bit more stable. And ironically, it's the A117V mutation that, if anything, is more similar to the silent prions in its size and stability, but is associated with disease. So sometimes in science, you start with this idea that, oh, if we figure out we can connect the dots, A to B to C, and instead we have A, X, and an exclamation mark, and I don't know, an ampersand or something like that. So it's always humbling, but I think it's important to recognize that we only have what's in front of us to look at, the fragments, and we're trying to do a lot of concluding from the fragment analysis. But again, it just tells us there's a knot of some shape, some size, but it doesn't actually tell me what type of knot is underneath that. And I'm not a sailor, so I can't give a better analogy of subtypes of knots. And it means we need to keep probing. I will end by mentioning we actually are going back and have now acquired a bunch more normal brain to really try to concentrate the silent prions and look at them in much more detail to see if we can learn a bit more. The challenge we've had is they're at such low concentration, it's really hard to find. So I will wrap up there again with sincere thanks to the donors and families who make this possible. A nod to Satish and Leonardo, who really are the drivers of this research in my lab. And I invite you to check out my TED Talk, and I'll see you later.
So I think we can open the doors and let our friends from room B back in, if any. And I know Dr. Watts will want to call his panel up for the Q&A, and while he does that, I wanted to make a plug for the book signing that'll be happening in the lobby at lunchtime. So David Langen, you're there, okay. David wrote a really nice short book about the caregiving phase for his wife, and I think it'll resonate with all of you. I would say you could read it on the plane on the way back, but I don't want to predict what any delays might be. So it's short enough that on an average plane flight, you can read a lot of it. Secondly, Don Friedman over there will be doing signings of his book called Still Phyllis, about caring for his sister Phyllis. And I want to say that Don has not only donated the books that he'll be signing, but he's donating the proceeds of the sale of his book. So please stop by, visit, take a look at those books. And I think, Joel, you want everybody to come up from your panel right now? Yes?
Yes, all researchers from panel A,
Yeah.
please join me up on the stage for the question and answer period.
So while they do that, one last book plug. Amanda Kalinsky is not here this weekend. She's recovering from hip surgery and sends her love, but she pre-signed some books, and they're out there as well.
All right, I'm going to start this off by giving a very brief, meaning maybe 30 to 60 seconds for each applicant, an overview of the main findings from their research so that those of you who are in the other room can have an idea of what this discussion is going to be talking about. And so our first speaker was from Jason Bartz, who was using the RT-Quick technique to examine the efficiency of decontamination of prion-contaminated surfaces. And so this is important because there are now known occupational cases of prion disease, or cases of prion disease that resulted from occupational exposure. So we need to have ways of validating that the techniques we use to sterilize and decontaminate instruments or our laboratory surfaces are actually working. And so what his research has shown is that bleach is a very effective method for decontaminating surfaces, whereas the common sterilant that one would use for microbiology, 70% ethanol, is not. So unfortunately, when you drink your beer, you're not sterilizing yourself from prions. Also important take-home message is that it's important to keep your lab clean because dust and other forms of dirt can interfere with the RT-QuIC test. So I think that's something that's applicable throughout science. Our second talk was from Professor Parmjit, who was tackling the problem of can we propagate human prions in cultured cells. And this is important because cultured cells infected with human prions may be a very, very useful tool for identifying therapeutics that can prevent prion disease in humans. And so his work has shown, and he said 20 years of work to get to what he's talked about today, where he took genetically engineered mouse cell lines where he replaced the mouse prion protein with the human prion protein, and then has done a lot of work infecting these cells with variant Creutzfeldt-Jakob disease material, and shown that these cells can be rendered extremely susceptible to variant Creutzfeldt-Jakob disease. That is a proof of principle. Once you infect these cells, you can get rid of the prions using an antibody. And that he is currently working to do similar cell systems for sporadic CJD prions. Next, we had Dr. Qiuye Li, who is the inaugural recipient of the CJD Foundation Postdoctoral Research Award, and he is tackling the important problem of what is the structure of human prions, or sporadic CJD prions to be more specific. It's not a given that the structures of mouse or hamster prions that have been solved so far are going to accurately reflect what the structure of human prions might be. And knowing these structures might be very important for when we're trying to develop perhaps small molecules that can go attack the prion structure. And so far, he has been successful in purifying prions from the brains of patients that have either the MM1 or MM2 subtypes of sporadic CJD and doing some preliminary characterization on these samples using a technique called atomic force microscopy, which can show you sort of the surface shape of these purified prions. He's a little bit further along in his work on solving the structure of chronic wasting disease prions. And very interestingly, it seems that these prions are going to have a different structure probably than mouse or hamster prions, which gives even more motivation for why we need to solve the structure of the human prions. Fourth was to my right here, Dr. Robert Mercer, who is using a novel technique called micromapping with the goal of trying to understand what proteins interact with the infectious version of the prion protein, PrPSc. And I like the term guilt by association principle. He uses something even better. He says by looking at the company they keep. So trying to understand which proteins a protein associates with might give you a clue as to what they're doing. And so he's using this really exciting technique that's only been developed recently to try and understand what proteins do PrPSc bind to. And maybe this is going to reveal molecules that are important for prion toxicity or prion replication. And he's currently at the discovery phase of his research, showing in principle that this system does work and will identify known interactors when applied to PrPC. So now he's applying it to PrPSc. And then last but not least, Dr. Valerie Sim is looking at the idea of the origin of prions. What are the very earliest events that happen in the brain, the earliest misfolding events that cause PrP to adopt an abnormal shape? And she used the term silent prions because these are misfolded forms of the prion protein that aren't necessarily on pathway to causing disease. And so she's looking at some of these potentially earliest forms of prions in brains from people people who didn't have prion disease, from healthy brains, and comparing those to brains from people with atypical types of prion diseases like the V180I genetic CJD or VPSPR or A117V GSS. And she's been applying her naughty knot technology to understand how different sizes, different shapes, different stabilities of prion aggregates might influence whether or not they progress on a path to become infectious or stop there and don't become infectious. And so that's sort of a general overview of what was funded and what was presented today. I would interestingly like to mention that I just published an article called The Importance of Prion Research that I wrote with some graduate students. And 4 of the 5 projects are 4 of my 8 unsolved mysteries in basic prion disease research. So I think that speaks to like the relative importance of the research that's being funded by the CJD Foundation. And so with that, I will open the floor to questions, and I'm sure all of the applicants or the recipients of the grants will be happy to answer them.
Well summarized,
Thank you.
Thank you. Great presentation, so thank you for sharing your results. Most of you talk about specific technologies in your presentations, from micromapping to IFM and others, right? So I'm curious if any of you have a particular technology that has been developed that could be a game changer, right? Like recently there was this AlphaFold breakthrough of being able to predict the structure of proteins. Is there anything in the work, in the technology side, that could be revolutionary for understanding prion disease?
Well, I mean, I think I have something to say about that. I mean, people have talked about cryo-EM a lot today, and I think as Val pointed out, 4 years ago, we didn't know what a prion looked like. We all had an idea, of course, but these structures that people have been showing, these cryo-EM structures, these prion fibrils, that's a game changer right there. And so the advancements in cryo-EM, I think, they've changed everything.
In my talks, I used to use a plush toy model of a fibril, and that's it. So
Yeah, everybody needs to hit the button to make your light go green.
I was just being funny.
So I'll comment on that too. So as a microbiologist, an analogy is, 15 years ago with whole genome sequencing. So in our graduate students, they would have a project, and being graduate students are like, well, we should just sequence the genome of this. And it's like, well, we can't. It's not feasible. It's not cost affordable. Now you can do it. It's super easy to do. And I kind of see cryo-EM as at that phase. Is right now it's difficult. It's very expensive, but as the technology develops, hopefully 5 years from now or so, it's going to be very, I don't want to say routine, but much more accessible. So I do think that's a game changer that way.
I think I cannot keep silent since cryo-EM is being mentioned so many times. So there's a new technology called cryo-ET, which T stands for tomography, which allows you to look at prions within cells. Remember how difficult it is to purify prions from neurons, and then if we culture neurons and then introduce prions, we will be able to see those at their native state, which will be a game changer in both structural biology as well as in prion studies. We do have some interest developing some device to culture those primary neurons to make it suitable for cryo-ET studies. This is quite an active field. Hopefully we will see some new results in the next couple of years.
Push it. There we go.
Let me make a push for machine learning. I think one of the things that I talked about was that we've done whole genome sequencing for 22 cell lines, and I have done RNA-seq on 88 samples. And I'm sitting here saying, well, we have the data, but we don't know what it means yet. So I think I'm going to push for machine learning, AI, and how to basically take data and make sense of it, such that then people like me can go back and ask the question, what does it mean? Can we do CRISPR-Cas knockout? Can we do causal effects? So I think machine learning and bioinformatics is what I'm going to push for.
And I guess I'll just add that it's an excellent question, because in my history in the prion field so far, each time there is a fabulous new discovery, it's been because a new technology's come along that opens the gateway. I mean, I was using field flow fractionation back in my postdoc to identify the most infectious particle, because now we could separate them efficiently. RT-Quick has come along and expanded PMCA to amplify infectious prions. And each level, and then the cryo-EM and the ability to handle huge data sets, CRISPR to knock out targeted genes. I think the delivery of medication, too, coming from a therapeutic angle, will be a game changer, because, of course, a disease that affects the brain, how do you get your drug or your target across the blood-brain barrier? There are novel, new ways now to do that. So I think it's actually incredibly exciting and hard to predict what the next big thing will be, other than to say there will be a next big thing.
Can I just add one thing here and show my age? When I was starting grad school, take it from me, restriction enzymes were not commercially available. So in fact, you could actually publish a paper where you'd purify the restriction enzyme and you do a restriction map with one enzyme and you could publish in like Nature. So this is where we've come in 40 years. So there'll always be new stuff. So I think that, but that's in my lifetime.
Hidden back there. My question relates to Dr. Bartz's presentation, and it has to do with the disinfection of the surfaces. And my question is, I'm not addressing instruments that are used for brain surgery and that sort of thing. I'm just going back to the surfaces. So where water and the ethanol did not kill the prions, what danger is there from this tabletop having disease-causing prions on it? If I touch it and I lick my fingers, is that a danger? If I touch it and I go pick my nose or something, or dig earwax out, is that a danger? So it's not that I'm trying to say we shouldn't have clean laboratories, but I'm just, I've always, through these years that I've been involved, thought of it as it's got to touch brain matter. Like through instruments or it's spontaneous.
Yeah, no, that's a fantastic question. And so right now I'm not, maybe my clinical colleagues can correct me on this, I'm not aware of any sort of transmission that's from what you described, touching of a surface like that. Yeah. Our kind of part of the motivation for developing these is that if you can minimize exposure, no matter what it is, it's good. Because to your point, there could be kind of weird situations where maybe you do touch your hand and there's something on the surface and then you have a puncture wound through that. So you can imagine all sorts of weird things happening. And so, again, working in a basic science lab, for as long as I've done this, we've used various things to decontaminate the lab with either bleach or LPH or sodium hydroxide. And we've always assumed that those surfaces were clean. And so now we can go back and do our normal decontamination, come back and use this technique and then say, oh yeah, they really are, or no, we missed a spot. And so I think that's an important piece of information.
And I guess the reason I ask, I'm an accountant, I'm not a laboratory person, but there's especially sporadic families who've had sporadic in their family member, you're always going, why? Why? What did, they didn't eat contaminated meat, they didn't eat wild game, whatever. But what if there was somehow on the tabletop in your kitchen, prion, the disease-causing prions from that family member got on your kitchen table or your kitchen counter, and you're going through and you've got a little cut on your finger, whatever. We're always asking why. And so that's what got me thinking about, is the touching of a prion, a disease-causing prion, really a danger to anyone, lab workers as well as family members? If it's just touch and it's not, obviously we can't touch our brains, but anything that could get up into it.
Yeah.
And I think I'll comment on that too. It's a very valid question and one I encounter a lot from patients. And actually, especially with CWD now, I get people asking me, well, if the CWD prions are now in the soil, or, what if I brought something, what if I'm washing my vegetables on the table? Do I need to bleach my kitchen? And all of this level of question. We can certainly go back to the epidemiology, and we don't see, it doesn't spread through households. It doesn't manifest that way. We can't draw that type of association. So that's one really good piece of evidence that it is not crawling on the tables, for example. I'm also reminded of this photograph where I did my postdoc at Rocky Mountain Lab with researchers, I don't remember from how long ago, but research precautions were different, I guess, back in the day. Literally, they had some prion brains sitting just open on the counter, and they were just standing around talking with cigarettes in their mouth, no gloves. And as far as I know, none of them succumbed to the disease. Now, we don't recommend that practice anymore. But again, it it's not something that we see transmitted, or it's not infectious in that way. It's transmissible if you inoculate yourself.
Right.
But it's not infectious in the way we think of, like, getting something and wiping it on your face.
When my husband was still, alive right before he died, and my son, who had been doing a lot of research on the internet, so that makes you an expert, my husband had a tear or something by his eye, and I went with my finger and just wiped it. And he goes, "Mom, don't touch his eyes." And I'm like, where are you getting this theory from? But it's the internet. So, but it just leads to that other thing of, touching, anything that they touched or whatever.
Yeah, kissing, sexual intercourse, anything like that is not transmitting this disease. So you're safe.
So I think cuts and abrasions is really what you're saying, right?
Yeah, but it's a cut like where you inoculate yourself with, for example, you're working with high titer CJD or BSE samples. This is also not inoculating yourself with tears. Spinal fluid has been a big question for us in the clinical world too, because do you have to treat spinal fluid as infectious? And there's been a lot of fear in labs. I've had a biochemist tell me, we can't put this killer fluid on our machines. Like, what do you, so even within the, the healthcare communities, there can be a lot of misinformation where we know that CSF is incredibly low titer. It is not, like, I wouldn't suggest inhaling it, drinking it, or inoculating yourself with it. But again, it is considered extremely low risk. The trouble becomes low and zero, right? What is zero risk versus low risk? But it hasn't caused disease.
How low is the titer reduction?
How low? I mean, you can detect it with RT-QuIC, right? But there was the Qiuye lab recently they inoculated, I think, 33 humanized mice with CSF samples from 7 patients, I believe, 4 to 7 patients. None of the animals came down with disease. So there was a prior study done where some CSF collected after death was able to transmit low-level disease, but it's suspected that may have that's post-mortem and sort of collected not by a spinal tap, but like sort of from around the brain, so it may have been contaminated. Yeah.
I guess to segue on that for Dr. Bartz too, in the decontamination, you said you work with University of Minnesota, and I'm from Minnesota. And when my wife died, I wanted to do the gold standard, like Ryan talked about, and have her autopsy done, but the Mayo Clinic refused to take her body. And the closest place I could get it done was Cleveland, and she'd have to be driven. And so I refused, and we didn't do it. So with your research and findings, will that help hospitals and clinics to do such autopsies?
Yes, I'll defer to my clinical colleagues for the autopsies, but again, as a basic scientist, so we do a lot of work on how prions are transported throughout the body, and we do a lot of microscopy, and we use core facilities within our university. So we use this technique in our core facilities to confirm that the facilities that we have used, we have not somehow contaminated them with our laboratory experiments. So we're using this right now in a lab setting. Whether or not this could be applied to what you're describing in a clinical setting, that I'll have to defer to people that know more about this than me.
We have time for one more question. Is there a question? Someone? No?
I think you guys know when lunch is starting. So those were fantastic questions. Thank you so much, and thank you to our panel. We have 55 minutes for lunch, followed by a beautiful photograph in the lobby with your purple t-shirts. So please stop and buy a book, have a book signed, enjoy your lunch, and we'll see you back here soon.